Okay, so bacteriophages, as mentioned, only infect bacteria. Why is a virus that infects a bacteria important for us to know about? And so they are super well studied because there's some important applications in understanding human health. Understanding the ways our bodies interact with bacteria. And we also have applications for medical treatment. So actually trying to improve some health outcomes. And there's lots of applications for using bacteriophages in research, right? Bacteriophages are all over us. So we have hundreds, thousands of different bacteriophages inside and on our skin. The exact kind of importance or function of having this um, isn't fully understood, but it's possible that the bacteriophages are actually function to protect us from infection with harmful bacteria, right? They could be helping to keep any bad bacteria from getting to us. Um, and so this is kind of consistent with how what we know about or our history of bacteriophages. So how they were initially discovered um, is there was a researcher studying dysentery. There was a patient with dysentery, um, and this uh, this sample from a given patient cleared up over the evening. So living bacteria is going to look kind of turbid, have a more of a, I guess, translucent quality about it. Whereas once it's dead, it's very clear. And so this researcher had a sample from a patient with dysentery, and overnight, rather than growing more bacteria, the bacteria seem to have all died off. At the same time, the patient recovered. And the thought is that this phage inside his body, which cleared the infection in this, in the tube here, also cleared the infection from the patient. And so if this is what happened, the bacteria phage we could maybe even think of as a helper to our immune system, right? Okay. And so I guess kind of next step here is this idea of phage therapy. So actually treating people. So rather than just relying on bacteriophages that may already be present, finding bacteriophages that can be used to stop life-threatening bacterial infections, right? And so you're likely familiar with the antibiotic resistance crisis. So a lot of bacteria are resistant to the medicines, the antibiotics that we've been using. Um, and so this would give us another tool, because the phages, can kill the bacteria. And so we know this has been done in mice. So treating mice with a bacteriophage one day after injecting them with a lethal bacteria allowed 60% of the mice to survive. And this is a lethal bacteria. So if not given the bacteriophage, 100% of the mice would die. Um, and it's being tested in some with human patients and some clinical trials, and is showing some promise, but these are still kind of early stages. So we'll likely kind of learn more about this in the years to come. Um, on the other side of things, um, the bacteriophages may, or we know that bacteriophages play a role in making some bacteria that infect humans, infect others, actually more toxic, more dangerous. So if we think about what we just learned about the lysogenic uh, cycle, um, a bacteria that's infected with a phage has new DNA in it, right? It has extra genes. And so there's types of bacteria that are avirulent, so the bacteria itself wouldn't cause harm to people. But after they've been infected with that phage and that's incorporated itself into the bacterial chromosome, um, those genes, some of those genes make toxins that are very harmful, and it can make the bacteria essentially more dangerous, or make it dangerous when it wasn't before. Um, and so an example of this um, is the cholera toxin. And so this is the cholera bacteria. It infects the large intestines. Cholera infections cause massive diarrhea, which can cause dehydration and death. It can be particularly dangerous in areas that don't have access to kind of clean drinking water, or, medical treatment during an infection. And so it's what we know is that the toxin, so the actual toxin that causes diarrhea, um, is in the middle of a prophage. And so the genes for the toxin are from the bacterial phage, not directly from the cholera bacteria. Um, and it's thought that this becomes kind of an advantage to the bacteria and the virus together. So it's advantageous to the bacterial phages kind of reproduction to be part of a bacterial cell that's doing well. Um, and the causing the diarrhea is essentially a way that is going to spread the bacteria to, new hosts. Um, okay, and so this Vibrio cholerae has both avirulent, so non-disease causing, and virulent strains. And without that uh, cholera toxin gene from the prophase, it's avirulent. Um, it's not going to cause issues for if someone's infected with it. It's only if they're infected with um, the virulent strain, which makes that cholera toxin. And here's some more examples. So bacteria that become virulent, they pick up toxins because they've been infected with a temperate bacteriophage. And so we have cholera, E. coli. So E. coli is a fairly common bacteria, but there's certain ones that are way more harmful to human health, and that's due to the toxin from the phage genes. We also see it with botulism, diphtheria, and strep. So strep, like strep throat, can lead to scarlet fever, and that is again due to the a phage. Um, and then very briefly, this, you don't need to memorize any of the details or know the details about this, but it is a very important thing just to know in general about the importance of bacteriophages. And so there's been a lot of research done using bacteriophages. So you may remember the Hershey and Chase experiment where they used bacteriophage, a T4 bacteria phage in particular, and they were able to discover that DNA is genetic material, right? That's a um, pretty huge finding in the world of biology. Um, it's been used in studying the regulation of gene expression, including the discovery of mRNA. Um, and studies with T4 led to the understanding that the genetic code was in triplets. So the three nucleotide codons. Um, okay.